Good afternoon, ma'am.
I'd already told you, children, we have one very small topic left in the chapter, and that is interesting facts about enzymes. It'll hardly take us 10 minutes for this, and then the chapter will finish. Let me enter these children. Okay. How many are there for the class today? Oh, I was not expecting so many. Since morning, hardly there is any strength in the classes. Interesting facts about enzymes. Just read the first fact. Let me explain it to you very quickly. Last topic it is hardly 10 minutes, beta. We'll revise quickly. After this, 5 minutes, I'll take your attendance. Just the first part. I'm moving on. Let me start. After everybody is admitted, then I'll start. See, the first and the most interesting fact about enzymes is they're mostly secreted in the inactive form. When an enzyme is in its inactive form, it is called as a proenzyme. Okay, and most of the enzymes, they are always produced or they are always secreted in inactive form. So first they are mostly proenzymes and later proenzymes convert into enzymes. So what is the inactive form of an enzyme called?
Proenzyme.
Proenzyme. And the active part, when an enzyme becomes active, we simply call it as enzyme. So most of the enzymes, they're secreted in which form? Beta, proenzyme or enzymes?
Proenzyme. Proenzyme.
Yes. Okay, let us understand this very simple. See, beta, let's talk about pepsin. You know about pepsin, it digests proteins. Now, supposing it was secreted in active form only in the stomach, then what would happen? Pepsin is not secreted as pepsin, it is secreted as an inactive form, pepsinogen. Okay, it is the proenzyme of pepsin is pepsinogen. Pepsin is not secreted in active form, it is not produced in inactive form. It is produced in inactive form, that is the proenzyme is called pepsinogen. Now, imagine what would happen if pepsinogen was secreted as pepsin only. The pepsin needs what? What is the food of pepsin? Proteins. You know pepsin is involved in the digestion of which nutrient?
Protein.
Protein. And if there is no food in the stomach, then what will the pepsin do? We know that the walls of the stomach, they contain proteins. The walls of the stomach, they contain proteins. So pepsin, it will start digesting the walls of the stomach only. So there would be a lot of problems. So it starts, pepsinogen remains in the stomach as pepsinogen till there is no food in the stomach. There are no proteins in the stomach. As soon as you eat food, you eat a food which contains protein. Then pepsinogen converts into pepsin because it has got the substrate on which it is going to act. Had not this been the system and a pepsin was secreted in the normal active form. So supposing you are not eating, you're fasting, or you're eating a food which does not contain protein, pepsin, what is its function? It has to digest proteins. So it will start digesting the walls of the stomach only, because the walls of the stomach contain what? They contain proteins. So it is better that the secretions are mainly in which form? They are in inactive form. So pepsinogen remains in inactive form. When food comes into the stomach, pepsinogen is converted into pepsin. As just a simple example to give you, to tell you that most of the enzymes are secreted in which form?
Inactive.
Inactive form.
Inactive form, and the inactive form is known as
Proenzyme.
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 Moving on to the second interesting thing about enzymes, some enzymes have different molecular structures, but they act in the same reaction. Some enzymes, when we talk about the structures, they are a little different from each other. Molecules are a little different from each other, but still they act in the same reactions. Such kind of enzymes are known as isoenzymes. What are they called? Isoenzymes. For example, we have lactic dehydrogenase. This is the name of the enzyme I'm telling you. Lactic dehydrogenase. We have 5 types of lactic dehydrogenase. I mean, the molecule, all these 5, there's a slight difference in the shape of the molecule of these lactic dehydrogenase. But all the 5, they help in the reaction. They convert what to what? Can you guess? Pa to La means what?
Pyruvic acid to
Pyruvic acid
lactic
to
acid.
lactic acid.
Yes, pyruvic acid into lactic acid. So this is the name of the enzyme. It is lactic dehydrogenase. There are 5 different forms of lactic dehydrogenase. And when I say 5 different forms, they differ in the slight structural difference. बहुत थोड़ा difference है between all 5 of them. All 5 are lactic dehydrogenase only. And they are all involved in the same process, that they convert pyruvic acid to lactic acid. So what is the second interesting thing about enzymes, beta? Some enzymes, they have different molecular structures, but they act in the same reaction. Third one, rennet tablets. Has anyone heard about rennet tablets? RENNET, rennet tablets.
Yes, ma'am.
Okay, where did you read about them, beta?
Ma'am, these
What
are
do you-
obtained from the stomach of the cows, which is used for the fermentation of milk to curd in the industries.
Hmm, good. थोड़ा सा, but milk to cheese. Huh? Very good. Basically, milk to curd means liquid milk to solid. कुछ भी हो सकता है, curdling of milk भी कर सकते हैं हम इसको. Excellent, बच्चे. Just remember that this is calf. We extract it from baby cows only. Okay, beta. Chaliye, let's study about rennet tablet. Very good, very good. Rennet tablets, RENNET, rennet tablets. They are obtained from the stomach of the calf, okay, or the gastric juice of calf. There are 2 options. Either we get this juice from the stomach of the calf, or we get it from the gastric juices of the calf. Now, in the stomach of the calf, we have an enzyme renin. The name of the enzyme is renin, and the function of renin is it helps in curdling of milk. So we have in the stomach of calves an enzyme which is called renin. The function of renin is it helps in curdling of milk. Basically, milk to curd, yani liquid to solid banane ke liye. Renin, what renin does exactly what your friend told you, that what renin does in the calf stomach, it changes the milk into curd. But commercially, हम लोग क्या करते हैं बेटा with renin? We use these tablets to make cheese. We use these tablets to make cheese. Gastric juice we take of calf. We take the gastric juice of calf. And from the gastric juice, we make these tablets. These tablets are added to milk, and then milk converts into cheese. What are these tablets known as?
Rannin.
Rennet
Rennet.
tablets.
They're known as rennet tablets because they contain that enzyme. What is the name of the enzyme?
Rannin.
Renin.
The milk changes to curd in the stomach of the calf. Basically, वो liquid milk को solid में convert कर देता है बेटा renin. And we take out the gastric juice of calf, or from the stomach we have some saliva part also, and we convert, because it contains the enzyme. Which enzyme is present in that?
Rannin.
Renin.
Yes.
Rannin.
So we make tablets artificially, which are known as rennet tablets because they contain renin. So when you add to the milk these rennet tablets, Commercially, your milk will change into cheese. So in commercial industry where cheese formation takes place, we use this enzyme renin. Okay, remember the name of this enzyme renin. Now we move on to the next one. Very small it is. Turnover number. This is a term often used for enzymes. What is turnover number? The number of substrate molecules acted upon by one enzyme molecule in a minute. In 1 minute, how many one enzyme can act on how many substrate molecules? When I taught enzymes, I had made that shape of E. And उसमें कितना substrates add हो रहे थे बेटा at a time?
2.
Two active sites, na? There were two active sites. So that means turnover number of that enzyme will be 2. Okay? Supposing I make a bigger E जहाँ पे 4 blank spaces and 4 blank spaces पे substrate add हो रहा है. So turnover number of that enzyme will be how much?
4.
4. So in 1 minute, okay, ध्यान से देखो, one enzyme molecule, only one molecule of enzyme in 1 minute, it can act upon how many molecules of substrate? That is called its turnover number. There is an enzyme, carbonic anhydrase. Okay, have you heard about this enzyme? Hmm? Anybody's heard of this enzyme anywhere?
Yes, ma'am.
Where, beta?
Ma'am, in recent class only you told that it is the fastest enzyme.
Yes, it is the fastest enzyme. Very good. If you remember, in 1 second we had made a lot of copies,
Yes,
and in
ma'am.
1 hour kitna ho raha tha? Exactly that enzyme. Too good. Carbonic anhydrase. This is your fastest enzyme. Okay. Its turnover number is 36 million. The turnover number of carbonic anhydrase is 36 millions beta. So you understood what is a turnover number? The number of substrate molecules acted upon by one enzyme molecule in 1 minute is called turnover number. The fastest enzyme till now reported is carbonic anhydrase. Its turnover number is 36 millions. Now we are going to do which is the slowest turnover number, which is the slowest enzyme. We have this enzyme lysozyme. This is a bactericidal enzyme which is present in your saliva and it is present in the tears. We have this enzyme in our body, lysozyme. It's a bactericidal enzyme, means it kills bacteria. Where is it present in the human body?
Saliva and tears.
Saliva and tears. This is the slowest enzyme. Its turnout number is the slowest, it is the least. So which is the fastest enzyme? You just need to learn their names. Fastest enzyme is?
Carbonic anhydrase.
And the slowest enzyme is?
Lysozyme.
Lysozyme.
Let us talk about the largest enzyme. Largest enzyme, when we talk in size, it is catalase. And the smallest enzyme is oxidase. You also call it as peroxidase. Any one name you can remember. So चारों जल्दी से याद करेंगे. Fastest कौन सा है बेटा?
Carbonic anhydrase.
Carbonic anhydrase.
Slowest?
Lysozyme.
Lysozyme.
Largest बेटा?
Catalase.
And the
Catalase.
smallest?
Oxidase.
Peroxidase.
Peroxidase.
Catalase, peroxidase, both are found in peroxisomes. Both of them, they're found in peroxisomes only. Okay. Children, as far as your UT paper is concerned, last time, again, I'm reminding you, I have made 40 questions, all on Google form. All are one-word answers. There are no MCQs. Okay. You have to give answers on your own. They're all one-word answers. You need to give answers on your own. On an average, I have taken 10 questions from each chapter. You have how many chapters for UT beta?
4.
4.
Four.
So I have taken 10 questions on an average from each chapter. So there is nothing that this chapter is important, this is not important. ऐसा कुछ भी नहीं है. All of them have an equal weightage. 10 questions from each chapter, 40 questions, no MCQs, one word answers. And the final thing is stick to NCRT. Read NCRT line to line, word to word, and you'll be able to perform well. Forget about your notes as of now. Forget about your all-in-ones, nothing. All this, we take it when we'll go to subjective part, how nicely you're able to express yourself. But objective is very important because you're going for competitions. So objective part also becomes very important. I have not given options because in options, sometimes you people make guesses and the guesses go right. And then the score is all, I mean, it's vague. It's no point having such scores. So just read NCRT. There is nothing big. If you start reading one chapter in one day also from NCRT, they are small chapters. Just highlight them with markers, important things, and you'll be able to answer. Clear, everybody?
Yes,
Even
ma'am.
read the summary,
Yes,
even, even read the summary which is given in NCRT. Read the chapter, read the summary. You'll get a lot of main answers from the summary also. You'll get clues, important things. Everything is written. Read every line of NCRT when it comes to these 4 chapters. So this is all about interesting things about enzyme. This was the only topic left. This was mostly secreted in inactive form, that is proenzyme. I gave you an example of pepsin. It exists in inactive form, pepsinogen. Then we talked about isoenzymes. Same reaction, structure is different. Then we even discussed about rennet tablets. Commercially, they are used to convert milk into what?
Cheese.
We understood the meaning of turnover number, the fastest enzyme. We learned the name, slowest. Once again, fastest was?
Carbonic anhydrase.
Carbonic anhydrase.
And slowest is?
Lysozyme.
Very good. And the largest and the smallest enzymes are?
catalase and
Catalase
peroxidase.
and catalytic and peroxidase.
So from tomorrow, your Diwali break is starting. I'm just showing you the next paragraph. You have to just read it. By the time you'll read, I'll take your attendance beta. Okay. This is something I've already done with you, but this I picked up from your NCRT so that you can understand this part. Please read. I'll take the attendance. It's very easy. You've done everything with me here. Okay. Akash, you're there, beta?
Present, ma'am.
Good. Anshika?
Yes, ma'am.
Okay. Aradhya?
Present, ma'am.
Ariba?
Present, ma'am.
Ayush Gupta? Ayush Patel?
Present, ma'am.
Ayush Patel was there yesterday also, is there today also. Drishti?
Present, ma'am.
Okay, dear. Harshita? Not there. Mansi?
Yes, ma'am.
Okay. Mansi, did you speak to principal, sir?
For what?
No, it was not Mansi. Acha, it was Mahi. Sorry, some other girl, Mahi. She has not yet done her registration. I got confused between Mansi and Mahi. Sorry, dear. Prabha? Raghav? Ritul? Okay, Ayush Gupta is saying, please mark me present. Okay, we'll mark Ayush also present. Who else said present? Harshita?
Okay.
Okay, Ritul is present. Sanvi? Sanvi, are you there? What happened to Sanvi now? This girl was so sincere. What has gone wrong with her? Sanvi, are you there? My God, even her performance has gone down so much. And then she's not attending classes. Is she there, Sanvi?
No, ma'am.
No, ma'am.
No, ma'am.
She's somebody who had 97 point some percent in class 10. She was the school topper in Chikar International class 10. What has happened to her? Shimail? Srishti?
Yes, ma'am.
Soham?
Present, ma'am.
Okay, beta. Uditi?
Present, ma'am.
Vedehi? Okay, dear. Vedehi? Soni?
Present, ma'am.
Okay, बच्चे. Pragya? शेजर?
Present, ma'am.
हाँ बेटा शेजर आते रहना, आपकी अटेंडेंस शॉर्ट है. अनन्या? अंशुल? माही? Mahi, are you there? Aryan Raj?
Yes, ma'am.
Good. And where is that new addition, Vivek Raj? As usual, he is not there. Okay, nature of enzyme action. I told you each enzyme has a substrate binding site in its molecule. Highly reactive enzyme substrate complex is formed. We had done na beta, enzyme substrate complex is temporary. When this complex will break, two things are formed. Enzyme alag ho jayega, and what is the other thing which is formed?
Product.
Product.
Yes, very good. We did this, that this complex of substrate and enzyme is very short-lived, means it's temporary. It dissociates into its product and the unchanged enzyme with an intermediate formation of an enzyme product complex. We have a temporary stage over here, and then this is formed. Catalytic cycle of an enzyme action. First, the substrate binds to the active site of the enzyme, fitting into the active site. सबको याद है, we had made an E and substrates बनाए थे. They were
Yes,
binding
ma'am.
to the active sites.
Yes, ma'am.
Yes,
Same
ma'am.
thing with the diagrammatically what I explained. I'm reading out from your NCRT. First, substrate is going to bind to the active site of the enzyme. It will fit into the active site. फिर क्या होगा? The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Substrate fits, वो lock and key mechanism, you remember? Exactly fit हो जाएगा substrate into the enzyme. Third one, active site now is in close proximity of the substrate, breaks the chemical bonds of the substrate, and the new enzyme product complex is formed. You get a complex which is temporary, and last mein beta, again वही complex will dissociate, enzyme is released, and what are formed is the product. The enzyme is again free to bind to another molecule of the substrate, and then this cycle can begin once again. Isn't it? We have already done this or not?
Yes, ma'am.
Yes, ma'am.
We also did factors affecting enzymatic activity. You remember how many factors I discussed with you? 3 factors I did with you. What are the 3 factors? Tell me.
Temperature, pH, and
Temperature,
concentration
pH.
of substrates.
Very good. So activity of enzyme is affected by change in conditions. They include temperature, pH, and change in substrate concentration. Same चीज है बेटा, ये देखिए. Effect of change in pH, temperature, and concentration. If you remember, I told you. देखो बेटा ये. पहला pH है. Enzymatic activity optimum पे जाएगा, then decrease हो जाएगा. Similarly, we did this also. Temperature. With increase in temperature, of course, energy बढ़ेगा. It will reach its optimum. But after that, it gets denatured. So again, enzymatic activity is going down. This we had done substrate wala. Up to a certain level, it will keep on increasing till all the active sites are occupied. इसके बाद, even if you keep on adding more substrate molecules, the rate of enzymatic activity will not increase क्योंकि कोई भी enzyme active site खाली नहीं है. All your active sites are filled by substrate molecules. So even if you keep on adding more substrate molecules, the enzymatic activity will not increase. Concentration of substrate, I told you about inhibitors yesterday, children, you remember? With the increase in substrate
Yes.
concentration, the velocity of the enzyme reaction rises at first. Initially, जब आप substrate बढ़ाओगे बेटा, reaction बढ़ेगा. If you remember, I told you 10 substrate add करे तो 10 products बने, 20 substrate add करे तो 20 बनेंगे. But there will be a time when it will reach maximum. This is not exceeded by further rise in the concentration of substrate because the enzyme molecules are fewer than the substrate molecules. So there are no free enzyme molecules to bind to the additional substrate. All the active sites of all the enzymes are filled now. So even if you keep on adding your substrate, कुछ फायदा नहीं होएगा. Activity of an enzyme is sensitive to the presence of specific chemicals that bind to the enzyme. This also I had explained with diagrams. एक substrate bind हो गया और दूसरा क्या bind हो गया था आपका? Inhibitor.
Inhibitor.
You remember? All of you remember?
Yes, ma'am.
Yes, ma'am.
Okay. So this process is called inhibition. Okay? When an inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of enzyme, it is known as competitive inhibitor. दोनों की shape एक जैसी थी, E वाला shape enzyme था, एक active site में substrate चला गया था, एक active site में inhibitor चला गया. So there was a competition. Such kind of a competitor was known as which competitor? Which inhibitor, sorry?
Competitive inhibitor.
Competitive inhibitors.
Yes. Very good. So inhibition of succinic dehydrogenase by malonate. ये भी याद होगा आपको. I told you malonate acts as an inhibitor. Okay. So succinic acid, this process gets inhibited. Such competitive inhibitors are often used in the control of bacterial pathogens. We generally use them to control the effect of pathogens bacterial. I also told you about another inhibitor which is not mentioned in your books. Which one was it?
Non-competitive inhibitor.
Non-competitive. Non-competitive inhibitor and competitive inhibitor में क्या difference होता है बेटा?
Ma'am, in competitive inhibitor, there is no change in shape of enzyme,
Very
whereas in
good. Competitive
non-competitive
inhibitor.
inhibitor,
Yes.
inhibitor change the shape of the enzyme.
हाँ, because बेटा actually what happens is competitive inhibitor में तो inhibitor active site में जाके fit हो जाता है. But non-competitive inhibitor there is a problem कि जो inhibitor की shape होती है वो ऐसी होती है that it is not able to fit into the enzyme. तो वो जाके कहीं और bind कर जाता है enzyme में. It does not bind to the active site. It binds somewhere else. And because of that उसकी shape change हो जाती है. जब उसकी shape change होती है तो active sites की shape भी change हो जाती है. And because of which जो normal substrate molecules थे वो भी add नहीं कर पाते हैं. And immediately process metabolism stop हो जाता है. I gave you one very interesting example of non-competitive inhibition. जल्दी से बताइए जिसमें immediately reaction stop हो जाता है.
Cyanide.
Yes, very good. It is cyanide. Yes, it is cyanide. Okay. Now this I did with you yesterday. Giving you 1 minute. I think I've already done with you yesterday. जल्दी से देख लीजिए. Oxidoreductase, dehydrogenase, transferases, hydrolases, lyases, isomerases and ligases. याद है ना बेटा कल करा था? इनको हमने numbers भी
Yes.
दिए थे. What numbers we had given them? You remember?
Yes,
E1 to
ma'am.
E6.
EC1.
EC.
चलिए. Now you read this, these equations and मुझे बताना ये क्या चीज है, ये कैसे अलग से क्या दिखाया गया इसमें. I'm giving you 2 minutes. Please read about the classification and nomenclature of enzymes, then I'll scroll down. Beta, once you're ready, raise your hands, then I'll scroll down. 3, 4 और here. 2 बच्चे वो दोनों भी I'll show you. फिर पूरे 6 को एक साथ डिस्कस कर लेंगे. Just raise your hands. When I'll see कि सबका हो गया, then I'll scroll down. You can see 4 on your screen. Read about them and then raise your hands. When you raise your hands, I'll go down. Very few hands raised. We're reading about cofactors. Chale, 2 minutes more for
Yes, ma'am.
cofactors. Read about cofactors also, then finally we'll discuss. Why only 4 hands? Hurry up. Let me see how many now. Good, good. Ayush, both Ayush are left, Soham and Uditi. So let's wait 2 minutes for them. Hurry up, hurry up. How many now? Good. Ayush, Drishti, Soham. Chale, never mind. Let's see. You people make your teams. Come on, let's have a quiz. I'll hide everything. Let's have a quiz of what you have read. Can we have girls versus boys? Count how many girls are there and how many boys? Let's have 2 team leaders. Okay, volunteer who all want to be the leader, then you can select your people yourself. Okay, who wants? Come on, 2 team leaders I want. Then you can select your teams. As it is, this is the last part of the chapter, beta. We're done with everything, just a fast revision is needed. It'll be better. So many brains together. Anshika, would you like to be?
Yes, ma'am.
Okay, you can select your team members. And second person who would like to be? Come on, I want one more team head. Akash, would you like to be the head? No,
Ma'am,
not interested. Not interested.
no, ma'am, but I'm confused. Ma'am, in
Array,
cofactor.
never mind. You'll have your team to support you. You just select team members. One name Anshika will take, one Akash will take, one Anshika, one Akash. So they're selecting their teams. So answers and questions will be in teams only. Chalo, Anshika, keep start beta. Select anyone.
Uditi.
Chalo, Akash, select next. Beta bolte jao. One she will speak, one you select.
Ma'am, Shahir.
Are you a new student? Akash, you are new.
No, ma'am.
तो क्या प्रॉब्लम है बेटा? फिर तो आप सबको जानते हो. Okay, Shahzad is taken. Anshika, continue.
Shrishti.
Keep counting also
Ma'am,
how many
Ayush.
one has taken. Very good. Continue. Continue, Anshika and Akash. Hurry up.
Soni.